I'd like to first of all start by thanking everyone for joining. This is a really exciting area now in precision medicine, and a lot of us are really happy to be engaged in this and working forward together in a collaborative process here. So today I'm going to talk about FHIR Genomics and also about the FHIR Connectathon overview. The latter may be also of interest to those who are not actually going to the Connectathon, but are interested in actually practical hands-on knowledge in terms of using FHIR Genomics. I'd like to thank all the different organizations involved listed at the bottom, including partnership with the Undiagnosed Diseases Network over here at Harvard Medical School. Let's start kind of from the beginning here. The Smart on FHIR and FHIR Genomics has really been an effort that has been ongoing for a number of years and recently kind of got some recognition in the precision medicine space. If you take a look at the Precision Medicine Task Force that was put together by the Office of the National Coordinator, they came out with a report just this past fall in September, which actually mentioned and recommended FHIR as an emerging standard and one that's especially useful for transporting data. And they suggested to build on current work, which involved Smart on FHIR Genomics, and cited a paper that was published in July in JAMA. You can see in the top right-hand corner there. They also recommended that it would be a good idea for PMI, for the Precision Medicine Initiative, to have ONC, Office of the National Coordinator, investments in pilots for FHIR, for precision medicine initiatives. And we've seen a number of such calls recently. I think there's been some calls, for example, these RFAs, requests for applications by the NIH, when you're looking at some cohorts that they're looking at building, as well as a coordinating center, which actually specifically cite Smart on FHIR Genomics and FHIR Genomics as something that would be important, to have and to work with. So in this talk, I'll really be talking mostly about FHIR Genomics, and I'll mention a little bit about the differences between Smart on FHIR and FHIR Genomics. All right, so first, what is FHIR? FHIR may be new to a number of us, and it is a relatively, new emerging standard, and you can see what the acronym stands for up here. It's basically designed very different from the other types of standards that had been designed before by HL7 in terms of actual standards that you would use to communicate clinical genomic information or other types of information to and from the electronic medical record. So FHIR is not only for clinical genomics. In fact, it's really was first designed, in terms of just being able to communicate data in and out of EMRs. And what it allows for is a strong extensibility and flexibility. It's concise, and it has these easily understood specifications. It's really made to be human readable. It's made to be friendly for developers. And as such, it has a well-defined data model and API. And it's designed around these resources. Resources are these small atomic elements that can be used when you're transporting information. They might be, for example, a resource about a patient. A resource can be a diagnostic report. And there are these base resources that have been defined and are being defined. And then when you look beyond that, when there are use cases and special cases when you want to extend those resources, then there are things called extensions that you can do on top of the resource. If you have a number of different extensions that are for a particular purpose, then you can define a profile which can contain a number of different resources, and a profile may be then well geared toward a particular application. So FHIR versus some of the previous standards, for example, HL7 version 2, which is probably the most widely adopted right now. FHIR allows for really an increase in consistency, flexibility, and conciseness. At the same time, it doesn't suffer from some of the issues that V2 suffered from in terms of segments that kind of made it stray away a little bit from standardization. In terms of FHIR versus HL7 version 3, the big thing is a decrease in complexity. With version 3, it was based on the RIM model, had a lot of really nice and interesting abstract designs, but it was people found it hard to implement, and it hasn't been as widely adopted. And FHIR is geared to change that by doing a number of different changes, as we'll explore in a little bit. So let's take a look at the FHIR Genomics timeline over here. So if we look here, FHIR Genomics really got its first kind of push in terms of the draft standard for trial use version 2 in September. So very recent, when the observation genetics profile was added, so that observation resource can capture genetic type of information. Right now, we're obviously in December here, where we've added into the current build the sequence resource as well as 6 genetic profiles that were based on work that was done and integrated work from a number of different participants, from organizations across GA4GH, Global Alliance, HL7, eMERGE, Ignite, and so forth, and all that feedback had been taken into Smart and FHIR Genomics, and then discussed within the HL7 community, and then actually put into the current build so that it can be tested out, and kind of like a test drive, basically, during the Connectathon in January. There's a document that was sent out with the webinar link, and I can send out again to people who are interested and maybe didn't see that. We are thinking of this definitely as a stepwise process. So if we see here in September 15, and that's the latest version that is, in the DSTU2, is based on only the observation resource. So people can actually send genetic information based on these genetic observations and not use the diagnostic report, right? And then in the current build, what we're doing is we've expanded it to include these additional genetic profiles in the sequence resource. And so we are, in fact, planning for this stepwise approach, and the document actually has information about how that stepwise approach takes place in terms of the mappings, for example, between going from those who are doing the DSTU2 to the current build and into the future. So how was this standard developed? How is it different perhaps than some of the others that in the past have been developed, like version 3, that didn't get as much adoption? Well, one thing that's interesting here is that it's actually built on use cases. So it uses the domain analysis model, which was also developed within HL7, and is currently being updated. And which had a number of different cases, and 6 of them are shown here that were very carefully designed with the different stakeholders. And that document is available. You can find online, or feel free to email me. We'll go through one of these cases today, later on. But just to give you a heads up about that, about how this was designed. The other thing that's kind of unique, I think, about FHIR is it's really the first standard for electronic medical records that does a number of things. These are just 3 things that I thought of, as I was making this. It's really made to be developer friendly. So it uses the latest technologies for that. It's designed to use, for example, JSON, XML, RESTful APIs. If you have a web developer that's on staff, they should be able to, using the extensive documentation that is available on FHIR, which is automatically compiled, to use it. So it's not a matter of reading these complicated manuals and trying to find other people have examples. There are many examples already up there. It's Electronic Medical Record Consortium supported. There's already projects out there of electronic medical record vendors who have already announced that they are going to be using FHIR. They have FHIR app stores, and there's for example, this Argonaut project where they're working together in sync to be able to make sure that their implementations all work together. So that's kind of special, and that when you will write an app, for example, you'll know that it'll be usable in all of these different environments. Whereas before, you couldn't really write an app that could be used in many different environments. And it's really geared, at least for the genomics part that we're working on, I'm finding, a number of people are finding, it's really geared for precision medicine. It's being designed for the next generation of clinical genomics, and we'll see that in the next slides here. So, how is FHIR Genomics kind of composed? How is it designed? Well, as I mentioned, FHIR has these resources, and it has profiles that allow you for additional specificity for particular applications. If you think about it, clinical genomics is and can use a lot of the existing resources within FHIR, such as the ones listed on the left, except for sequence, which is a new one that we added and is in the current build, that is geared more specifically for clinical genomics, although it potentially can have some other uses. And then for each of these, there is a current profile or a future profile that may be, that is in development. And so there's one specifically for genetics that captures all the things, and we'll see a few examples in a bit for that lets you see individual observations, diagnostic reports that allow you to put together those observations into a diagnostic report, information that you want to capture about family history in terms of their genetics and pedigrees and so forth. And then the diagnostic order, perhaps if you want to place an order to a lab, what kind of information do you want to specify about the genetics? Sequence, since it is geared toward the genetics from the first place, is currently a base resource, and additional profiles will be able to take that and find individual cases that may be useful to extend that in the future. And then as an example, we created a very specific profile that looks at HLA genotyping results. If some of you saw the recent IOM digitized report, that gave an example of an HLA type testing that could be done, just came out a few days ago, and actually mentioned FHIR Genomics DSTU version 2. But we're currently working with some experts in this area of HLA genotyping for the Connectathon to really test out this profile and then see, how that works in terms of extending the diagnostic report. So what are the advantages of this approach? First of all, there's low, or at least there's no or low repetition. You get to reuse the existing FHIR resources, so you don't have to have an increased learning curve, and you can work together with all the other laboratory results that are done in FHIR, so you can easily combine, for example, CBC, blood counts, and so forth, with your genetic information, imaging information, and so forth. It's well organized in that you are able to localize the genomic and omics information in a sequence. So when you need that detail about sequence information, it can be localized within sequence. In a sense, it's a safe harbor for that information. So it hides that, it's a layer of abstraction that hides that complexity from the physician unless they would like to see it through an app, which can do that and access it. In addition, as we'll see in a little bit, the sequence resource allows you to point to repositories and other places that have more deep sequence-based information if you want to do further analysis. It's also easy to implement. I mean, I emphasized this before, but I think this is a really big one for why adoption is the way it is in terms of FHIR, is that it's really designed to be developer-friendly, uses the latest web technologies, so you don't need to pull out that COBOL programmer, from way back. And then consistency. All the genetic observations also use the observation genetics profile. So it works well with other observations that you make, so you can add that in using the same type of approach. So let's take a look at, for example, how one of these resources and profiles might be used. If you see here, the scroll here is an example of a resource, and the profile is encoded by this circle. So we've got this regular diagnostic order that comes with FHIR, then we add on top of that genetics type of elements, and then you can have a pointer, for example, to sequence when you want to place an order. Then when the lab responds, whether it be an external laboratory or an internal lab within the hospital, then you will get a diagnostic report. Again, it's structured to have similar information to a regular report, but on top of that, you have this additional genetic information that can be observations about genetic information as well as sequence information if you want additional detail. So what do we have in the sequence resource, and how does it leverage existing technologies? So in the sequence resource, first of all, you can, if you'd like, to include just a single variant piece of information along with key elements about that variant. Or you can ship along sequence information, perhaps at the gene level. On the other hand, maybe you have such a huge sequence, or maybe a number of sequences across several chromosomes that you'd like to point to. You can do that too by pointing to external repositories and take advantage of the deep type of sequence information that you might find, for example, in an NCBI or Global Alliance repository. So you can do that, and then people can access that information as well. It's also extensible to new types of omics that are all based on sequences. So when you think about it, for example, RNA-seq, again, based on sequences, gene expression, based on sequences where you're measuring the quantity and so forth, when you're looking at RNA-seq in terms of having essentially a digital version of gene expression, and many other cases as well. So let's take a little deeper look at this, and then we'll go through a few examples. So if you look at the sequence resource, as I mentioned, you can look at actual single variant piece of information, which you can encode as codable concepts. So you can identify the variant as being from ClinVar, dbSNP, and so forth, and then you specify the type of ID that was used. That's what the codable concept allows you to do. There are many others here. There's documentation that people can click around on. I don't want to spend too much time, but just maybe also tell you can also get information about the sequence. But you can send information about a sequence, and that's kind of very carefully defined in terms of starts and stops and so forth, so that you can reference that and include that in the information that you pass along. These are some of the other attributes. Some things that we added that a number of laboratories were interested in was the quality. Quality can vary at different regions within the sequence, and that was a metric that was added there, and so that people can get a measure of how well they can trust that information. Again, we're envisioning, and there are already some apps that use this type of information to decide what to show to the clinician versus what to show, for example, to a researcher. So it really helps to bridge between the clinical world and the genomic world. Here, as I mentioned, there's a way to refer to external repositories. And, just as an example here, I just show the GA4GH, where you can have you can go all the way down to the read group level, right? Physicians usually not going to be interested in that, but an app that wants to make do certain types of analyses may actually want to go to that type of level of information. So it can point to the URI, and then it can access that information rather than having a huge payload every time you want to pass along the sequence. So let's take a look at a few examples. What do these messages actually look like? What do they contain? That's kind of what we have in the next few slides. So we have here the diagnostic report resource, where it's been extended with this, with this genetics profile, looking at a particular type of a gene mutation analysis where we have a panel in this case. And you can see a number of pieces of information are passed along, a number of which are very relevant to, in this case, this particular condition. And then this particular test where you will have a LOINC code, which is a laboratory code that actually specifies that particular test, as well as SNOMED codes that, for what type of test it is. And then you'll be able to also encode in that report, the actual results with codes such as whether it's positive or not, what the location is, and so forth. All of that can be encoded here, and these can point to individual different observations. So the diagnostic report, there's a hierarchy, and the diagnostic report points to these observations, which have additional information. Here's a little bit of detail of another example with an observation resource, where we're looking at that profile with additional information. Here we're taking a look at a somatic example. We're looking at a panel, and there's going to be some somatic information that's going to be encoded, in this case looking at EGFR. So for cancer, there are a number of different panels that look at that, and this is a way that can be encoded again. And again, these can be components that have different pieces of information, for example, SNOMED codes to explain the malignant status, pathogenicity, and so forth, interpretations. And furthermore, you may want to actually communicate a sequence. So while there have been a number of, people been doing panels, SNP chips, and so forth, there are also next generation sequencing and deep sequencing of particular genes, and that you can encode here, for example, with a sequence resource, where you may have a reference sequence or transcript that you're going to be referring to, and actual coordinates, and then you're going to have the actual sequence and potentially a repository or pointer to the cloud where you're going to store the full larger sequence that it came from. So I think with that, I'd like to kind of also tell you a little bit about kind of where this is going in terms of the big overview. Then we'll stop a little bit for, in the midway point here to see, if we have any questions, and then we'll go on to the Connectathon period. So in this part, what I'd like to talk to you about is kind of how all these pieces fit together, right? So you've heard a little bit, about FHIR Genomics, Smart on FHIR Genomics. There's HL7, G4GH. How do all these pieces fit together? So this is one kind of rendition of a sequence of events that can happen, where you start here with an electronic medical record right here, which Smart on FHIR builds on. And there are a number of different app stores and so forth that have already been built by a number of companies that support Smart on FHIR to work with the EMR, electronic medical record. And Smart on FHIR contains FHIR, including, now the latest Connectathon build, HL7 standard version of FHIR Genomics, which is the version that's in the current build, and it will be test drived in the Connectathon, as well as some smart services. And those services are really that value added to be able to enable apps to work. And we'll see that in the Connectathon what some of those are, but they include things like authorization, privacy sharing, issues around launching an app to give it the context of what is going on in the electronic medical record for that patient, and scope, and issues around what that app can do and cannot do. And so what we have is an example here of what would happen. First, you would have a diagnostic order app, which might place an order, so it needs to have clinical data about the patient. And then it would want to ask for a genomics laboratory, for example, to make a genomics lab order. And so it will place the order. So this diagnostic order app is a Smart on FHIR app. And here's actually a drawing of it made by in collaboration with the Smart on FHIR Asia team. And an order is made to the Smart on FHIR Genomics lab, which also offers smart services, includes the HL7 FHIR Genomics, and for raw sequences and for repositories, can include implementations of things, in this case, it's going to be the G4GH Global Alliance format so that searches can be optimized. So all the raw data can be stored here, much of which people, and we've interacted with electronic medical record vendors, they're not interested in all of that raw data. They'd rather have a pointer to it somewhere else. So then what you get is an order response, number 2 here. Again, sent along with a FHIR, using the regular, order response, which is part of FHIR, to the diagnostic reporter app, which you can see here, again, built in collaboration. This is the Smart on FHIR Genomics app that gets that order response and saves the actual report to the EMR with all the information that we mentioned above. We saw a few examples of observations, diagnostic reports, and the sequence resource. Now, the information is back in the electronic medical record. We're back in the world of the physician and other healthcare allied providers and other individuals who are going to potentially access this data. They can run a Smart on FHIR app on top of that. And an example here is one that is was designed and built over at Vanderbilt University by Dr. Jeremy Warner and others. And in collaboration, they've designed the Precision Cancer Medicine app. This particular application is able to work together with Smart on FHIR to show your existing patient, for example, if they have an EGFR mutation, to show those panels and show you how your patient is similar or different compared to all the patients that are in the Vanderbilt system. And so that is an example of an application of how you might use the data that was obtained through this workflow here. So with that, I'd like to kind of, we're reaching approximately the halfway point. I wanted to give you guys some useful links. Again, as I mentioned at the end, you can feel free to email me if you'd like to get these, this presentation. But this will have, these clickable links that have, the version 2 of FHIR Clinical Genomics, People interested more in the deep sequences and how you might want to do repositories. I mentioned a few things about GA4GH. I've been involved in both of these, so feel free to contact me on these. I guess I didn't do an introduction, but I'm one of the I'm a co-chair on the HL7 clinical genomics work group, as well as on the executive leadership of the clinical work group of the Global Alliance for Genomics and Health, as well as on the e-health team there. And then, you may be interested to see the initial paper on this, which, like all papers, were obsolete probably just a couple of weeks after they were published. But in JAMIA, there's this paper, which has already kind of become relatively highly cited after the ONC report. It gives you a lot of background, which I think can be useful. And then just, a little bit about smart genomics. Maybe you want to see some of the apps, the smart platform, which is not just genomics, but an overview, and you can go there and click on the gallery and see dozens of different applications, not just in genomics, but across the different area here. Yeah, thanks for those who are interested. I already am seeing some chat messages for people. People are interested in getting a copy of this. If you could, yeah, send me an email, that'd be wonderful. Although it says FHIR Hands-on Connectathon, it's really about hands-on. Whether or not you can be in the Connectathon, we're going to give everyone who requested a virtual machine. So it will be like as if you are at the Connectathon. You have a virtual machine with the server, the applications, and then you can test out all of the stuff that we're going to do later today. And so I will show you some things that you can test out even without the virtual machine. As you know, the virtual machine is going to take up almost a gig of memory just to download. There's some things that you can try directly on our server as a client. We are at just slightly past the halfway point, so it's a great time to move on to trying this out. So this is one of my favorite parts because it either works or it doesn't. So no, this is the part that we actually get to try things out. So if you guys have a browser, for example, we recommend the Chrome browser for this particular demo because then you'll have the same screen that I have. And what you can do, everyone can see my browser now, is that correct? Right? Yes. Okay, perfect. Thanks, Kate. So if you take a look here and you go to this website, http://genomics-advisor.smartplatforms.org:2048 for the port number, you will be able to sign in and follow along with what I'm doing here. Just for your, we made a kind of a temporary email address, name@mail.com, and password is just password. If you do that, you should get to this screen. So once that means that you're logged in and you'll have access to in this case, the FHIR Genomics, the Smart FHIR Genomics API sandbox, because it also enables you to do apps and so forth and import data in that. But for the purposes of what we're going to do, once you log in there, then you're basically all set. So we're then ready, once you've done that, to go to the next slide here, where you can see that we're actually take a look and see an actual sequence. So we're going to see, what we have. So if you were to do an API call, it would basically be to get, and then the base resource that you're calling, the type, and the ID. So in this case, you'll see here local version. That will be the version that you'll be able to use if you use the virtual machine. In this case, we're going to use the online version. So if you basically just were to cut and paste this from this slide and to paste it into the web browser, you would be able to see this here, which actually shows you an actual sequence that that is obtained here. It actually includes a number of different sequences because we're just looking for all the sequences. And you can scroll down and see that in fact we have perhaps 100 or so resources here, sequences that are being given. And that is the number of results. So you don't maybe want all the sequences, maybe you just want one of them, right? So you are able to, by using the ID of the sequence that you want, to get a particular sequence And in this case, look, for example, for a particular observed allele value, and that information will be encoded in that sequence resource that you have. And again, if you just put this in your browser, this should work, as I just did before the meeting. And beyond that, I think one of the most powerful things about FHIR, and this is true about FHIR in general, we just add this ability for within FHIR Genomics to have these search parameters be especially relevant and optimized for genomics, is that you may want to do searches. So for example, I want to get all the sequences that have a particular variant ID. So this search will do that for you. And as you can see in this search, that the code value does have, this example does have that result that comes through. But, that's kind of very simple search. I think the beauty in terms of searches is that you can actually do partial hits. You can even do mismatch hits, which we're not going to go into here, but you can do searches based on different fields that we enable. So, for example, maybe you're looking for a particular species. You're looking for Homo sapien, and then just use, the regular type of thing that you'd use in a RESTful API here. Or maybe perhaps you're looking for a particular SNOMED identifier. All of those IDs, SNOMED ID, all those IDs can be used here when you're doing your search. So, just an example. Let's say you're looking for all of the drug-resistant tuberculosis results that have a particular variant. You can look for results that have that particular species and look for variants that came out of that. So it's very powerful to do that. So the next part I'd like to turn you to is the actual server. So as I said, I'm very happy to report that we have built a server that is, we're giving out, that code is open source. So it is up on GitHub. That server will be part of that virtual machine image, which is a Linux-based image, and will include both the code for the server as well as a number of examples. It can load examples for you. It can load the latest specifications. It can dump. It can set up the database for you and dump the data. It can submit data. Let's take a look at some of the more important files for those who are interested in, some of the finer details, and then we'll get back to looking at the top-level picture in a little bit. So, we support a number of different things here. This is a Smart on FHIR server, so it supports not just FHIR Genomics, but some of the Smart API things as well, and that's why all the calls first go to API, so Smart can intercept it. But then FHIR Genomics calls and FHIR calls go to the FHIR API. PY, these are all in Python. There's a Python FHIR parser that could be useful for anyone, even beyond this particular application. There's a way to index the data in a database. There are the data models, and a nice query builder that takes it from FHIR into actual database queries to use Postgres SQL here. So what does the FHIR Genomics server do here? So it allows you to parse the data. For example, is the data in the correct structure format, the correct types? Is there an error in it? It allows you to index data for, in the database. The database is part of that virtual, machine, and also commit structured data into the database as well as to respond to different client queries. And then, people ask, so what about the Smart Genomics? What does that do, on top of the FHIR Genomics? Well, really, FHIR Genomics, as I mentioned before, kind of essentially, the current build came out of the last version of Smart Genomics after a lot of feedback was added from different communities. But on top of that, Smart Genomics isn't about just transferring data. Which FHIR Genomics is about, which is a common way to transfer data to and from electronic medical records, especially genomic data for the FHIR Genomics. But the Smart Genomics is also about how do you actually make it usable by apps. So, and how do you even use it with existing systems? So how do you transform all that raw data you have out there? It might be from Illumina, 23andMe, VCF files, G4GH, all of those have transformations that are, part of the server here. The ability to store search-optimized GA4-GH and raw sequence data. That's an area that is part of the idea of Smart Genomics, the lab server, you know, is a part of the feature set that the Smart Genomics is working on. The ability to authenticate and authorize. We'll take a look at an example of this, which is, you know, part of the virtual machine. And the idea of getting context for apps, which is a general one in Smart on FHIR, but is one that's useful everywhere, where the app knows where it's been called from, so it knows what kind of information it is allowed to see, what information it already has because the physician is looking at it, and so forth. And then issues around privacy and sharing, which is really important around genomic information. So as I promised, let's take a look at a couple things in high detail. For example, here we have the API.py, you know, the Python code for the API call. You get, you know, first you get the actual call, you verify the access, you are able to process the request, and then you call, you know, to do the search, you know, to do the actual search if called upon. So it really handles, really at the top level, it hands it down to the next level. I think for more detail, I'd really encourage people to, you know, just take a look at the code. I just want to show you one quick example so that people can, you know, when you get the slides, you can kind of see where to start, and those files are the places to start. How do you run the server? Pretty simple. Just type Python, you know, server.py-d at the command prompt once you're in the virtual machine, and then once you start, once it processes calls from a client, you'll start to see what the calls were and so forth. So I think, you know, the file is big. I recommend people to send an email. We know we're going to get a lot of emails, so I'm asking people to please use this exact subject in your email. Please send webinar VM, so that way we can process all of them in a timely manner. And of course, the server, as we mentioned, is open source. It's on GitHub, but, you know, to install it, you know, you need to install all the different libraries, Postgres, SQL, you know, all that, and that's why we made the virtual machine, where you just basically you just hit go and it runs, right? So, and then for those who want to just try the client side and want to try some of the different sandbox things, you can just go to our online server and register with that temporary ID that I gave people to use there. And that is a way that you can move forward regardless of whether or not you actually go to the Connectathon. So finally, I'd like to take a look at, you know, an actual use case, you know, stepping back up to, you know, a higher level in terms of not looking at the specific code at first, but how do we bridge from a use case and go to the code? So we're going to look at use case 2, clinical sequencing, and we're actually going to take a look at cancer profiling, and we're actually going to look at a population and see, in this case, what we're going to look at is for this patient, which, you know, it's going to be named Fred Appen as this example. We're going to look at somatic analysis. We could also look at germline, but, you know, in this particular one, we're going to actually look at somatic, and this would be the call to get all the somatic type of information, observations, and observations, of course, contain sequences and variant information. So that would be the call to do that. If we want to do the call, you know, on that sandbox, you know, then you can actually make the call like this directly, you know, rather than doing it through code. Here's, you know, the result that you would see. And so basically, you know, it's this Atom feed where you have all of the different observations that you've been asking for. And as you can see, you know, in this example, you know, we see one here that has what we're looking for. We're looking for somatic, and it's for the particular patient that, you know, that we had requested based on the patient ID. So what does FHIR Genomics really accomplish for us? Well, it allows us to extend the existing FHIR, right? It extends it specifically for genomics and omics data. So it's not just for genetic tests, but it really allows us to bridge into omics, into sequencing, RNA-seq, you know, those type of omics technologies as well, and achieves consistency as well as being concise. We really try to design it so that there is few new additions as possible but at the same time, it allows for flexibility if you want to design a new profile that has a very specific use case, such as HLA. We looked at the HLA use case. Well, we didn't look at the use case, but we mentioned that that is an example that you could do, and we actually modeled how to do that, and we're looking at testing that, test driving that particular profile further at the Connectathon. This approach allows you to maximize relevance to clinical genomic applications by being built so that Smart on FHIR works very well right on top of this FHIR Genomics, right? And so these clinical apps have been designed, and we've got a number already. They're being built, I mean, literally around the world, multiple sites in the U.S., multiple sites in China, Europe, and other places. And feel free to email me if you'd like to start on that. We have also open source code for a number of these applications where either the users or we've developed them that we can share to help jumpstart applications. And then, of course, I think this is key, that rather than reinventing the wheel, we leveraged existing technologies, right? We worked together, and I'm working on extending now the domain analysis model. Just had a call this morning with some people actually at Vanderbilt, some oncologists who are going to be adding to the use cases and some new use cases so that we can develop this further. We leverage existing repositories, existing formats that are for deep sequences, existing types of work, whether it be done by ClinVar, working with ClinGen, those other types of consortiums that are developing approaches. And so I would say that right now, you know, although we have something and something that is being, you know, used and people are building apps and it's being tested, I think just as David mentioned, you know, this is really just the beginning. But I think it's a very exciting beginning. And so with that, I'd like to really invite everyone, well, as long as you don't overfill the room, to come to the Connectathon, which is currently, I know a lot of people have asked about remote, but so far it seems that right now they've been asking us to, for the FHIR Connectathon, to have people come, to ask people to come to Orlando if they're interested, because there will be limited bandwidth if everyone connects from outside. And the room has just been expanded from 100 to 200 people. January 9 to 10 in warm, nice Orlando, right near Disneyland there. Although I don't think we'll have much time for Disneyland because we'll be there all day. And we will have an opportunity there, everyone who's there, to learn from experts, experts in FHIR, experts in clinical use cases. There'll be physicians, there'll be developers, there'll be people who worked on FHIR, there'll be electronic medical record vendors. All of those have been confirmed, and those teams are coming. And you'll have an opportunity to tackle real clinical genomic use cases. And I can send you a link to the Connectathon if you'd like. I think that it may have been on the webinar link as well. There are actual use cases that have actually been written in there already that we will be going through. And we also have code samples that help people start on that. So even if you're not a programmer, you can take a look, and you'll be able to use like a web browser like I showed here, and be able to click and be able to see how an app would use this, as well as try out some of the new apps that people have been working on. If you're a developer, I mean, I think this is a great opportunity to get kind of your project out there in terms of developing apps, because we'll be putting apps that are developed from there into the app gallery. That is our goal, to be able to have apps that showcase, you know, how this works. And from developing the apps, we'll see what additional work is needed and what we can do to change things going into the future. You can look at the current app gallery by going to the Smart Platforms webpage. So with that, you know, I'd like to kind of point you to a few other places if you'd like to get more information specifically about the Connectathon. As I mentioned, you can play with it from home. It's just that you'll kind of lack I think the big advantage of coming to the Connectathon is all those experts will be there and all the different parties will be there to help, and so we can all help each other, because none of us is an expert in everything, and so we can work together. So if you look at the Connectathon snapshot by clicking on this link, if you look under the FHIR page, the directory, you'll be able to get the snapshot, the entire specification that we'll be working with in the Connectathon, which is the one, in fact, that our server that we give the virtual machine, it includes this specification. These are a few links if you'd like to look directly on the actual sequence information, sequence resource information, the diagnostic report for genetics, observation for genetics, diagnostic report for genetics. Don't have to copy this down immediately because all of these are actually accessible from this link. But this is, for those who have the, the PDF or the slides of this later, you'll be able to hopefully, quickly copy paste that if you'd like to. I think with that, we're hitting about the 10-minute mark, so I'd like to really thank, and this is just a very small list of just tons of people who have been involved, but these are people who have been involved in from, the early days here, whether it be Smart on FHIR itself, so tremendous help from all these individuals. I'd like to name a number of them from, David Creator, Rachel Ramoni, Josh Mandel, Ken Mandel, Isaac Hahn, there's tons more people. I wish I could name them all. And then Smart on FHIR Genomics, the genomics aspect, everyone from Hemming Yao, Tom Chen, Pei-Jing Zhang, Jeremy Warner, currently at Vanderbilt, again, many more. And then our most recent addition, the Smart on FHIR Genomics in Asia contingent, who have been developing a number of apps and are taking this to China, and it will be used there for a number of different interesting deployments and use cases, which are starting very soon. So there are a number of pilots and people that are implementing this now. I can't actually talk about, I'm not sure which ones I can talk about, so I will not talk about any, but just say that there is a lot of activity in this, and if you would like to learn more or be posted on, as pilots get announced, we're going to start to have a web page where people who are working in this area, we'll put you on that Harvard web page and/or the gallery to list your efforts as you would like them to be publicly announced, and we'll announce the ones then publicly that want to be announced. So with that, I'd like to close, include this email address that will always work, just sort of this permanent email address, although you can certainly feel free to contact me at my MIT or the Harvard email address. Again, if you want to get slides or the virtual machine or anything else, if you could kindly follow this format for the subject line, then your emails will automatically be categorized by my email client. It will be a lot easier to get back to you in a quickly manner. So with that, I'd like to close. So there are a couple of different technologies, for example, the TouchTone project by Aegis, which is Wildfire, I believe it's called. I can send you guys a link which helps to test conformance with a test resource. There will also be test scripts that are part of the Connectathon that will be posted on the Connectathon webpage. I think I'd like to really take this opportunity to thank you guys for all coming. It's really been great to be able to share this new technology with you, to be able to potentially get your feedback through this session as well as email. And once you try out the virtual machine and/or the website directly. And for those of you who are thinking about the Connectathon, maybe you're not sure or you'd like to ask questions about how that would go, feel free again to email me on that. So again, thank you very much, everyone, and we will try and post these slides and, if possible, a video recording as well. Okay, have a nice day, everyone. Bye for now.